Thank you for two very interesting talks. Please join me for the panel discussion. And John Taylor. John, do you want to join us as well for the panel discussion? No, you're good back there, all right? We have someone in the audience working on the ecology of fungi as well, fungi. All right, so questions, please. In the back.
So mammalian, like the parasite nematode, they do deliver small RNAs got taken up by mammalian gut epithelial cells. And then those parasitic small RNAs basically can suppress human inflammation and immunity genes. They do in a certain degree, to a certain degree. Oh, yeah. So that's actually very important to make sure that when you design those RNAs, you want to make sure they are not targeting any host genes or human and animal genes. So for I didn't go to the detail for the transgenic plants we developed to try to design fragments that can target 4 dicers, 2 in botrytis, 2 in verticillium. We try to avoid those conserved regions. So we eliminated the possibility that they can target any homologous genes in other species. So in that case, we have to design 4 different fragments to target each dicers. So the design is super important to avoid the off-target effect.
This question is to Dr. Naar. You talked about transcriptional factors in your presentation. Both the host and fungus being eukaryotic, how do you prevent toxic effect of this? Would there be any toxic effect? That's my first question. And then the second question is RNAi the solution to fungal infections?
Yeah, very good questions.
Using liposome, the liposome vesicles.
Sure, certainly. Yeah, to address the first question, we carefully selected the compounds that have basically a window of therapeutic efficacy. So IKIX1 doesn't bind effectively to the human KIX domains, and it's about 100-fold decreased affinity for the human KIX domains. So this is something we carefully screened to begin with to avoid any kind of toxicity. We put IKIX1, of course, into mice first for PK, PD, and also for tox, and we didn't see any toxicity. For the second question, yes, I think that's a very intriguing possibility that was alluded to as well. So my lab, the other part of my lab actually works on microRNAs, and we're very interested in therapeutic targeting of microRNAs and perhaps delivering small RNAs as a therapeutic for fungal disease could be feasible. I agree.
I had a question for Anders too. Have you tried your approach on Candida or other fungi that also have mutations that make them resistant to the antifungals in the ERG11 genes?
So yeah, good question. I should have mentioned, so there are multiple mechanisms that can lead to resistance. So you could have the target also being mutated, and that's common in Candida albicans, for example, or ERG11. The target of azoles is actually mutated. We haven't looked at that in particular. We looked at a number of Candida species that have intrinsic azole resistance, and we did see that IKIX1 has a function by itself in those, and we don't know what the mechanism is. It seems to be unrelated to the azole sensitivity. In those strains, we see not synergy, but rather additive response. So it looks like there's another function of IKIX1 that still needs to be elucidated that has an antifungal mechanism.
So my question is for Hailing. I think it's quite interesting that these extracellular vesicles from plants have some selectivity in the small RNAs that are encoded. So are these small RNAs, inducible during pathogen infection, or if you look at the repertoire of small RNAs, are they more likely to not target something in the host, but target something for a potential pathogen?
And so some of those small RNA, transported small RNAs are induced by pathogen, but not all of them. And then some of them have a host target themselves too, because like some of those TACI RNAs, they have their host targets too, but more than half of those doesn't have host targets. So now we kind of figured out, partially at least, how they got selectively loaded into vesicles. So we are writing that paper right now. So basically they are RNA binding proteins. There are several RNA binding proteins seems to contribute to the small RNA selection and the transport.
I have a question for Dr. Naar. In terms of looking at the multi-drug resistance of your PDR1, have you looked in Aspergillus, and does your small molecule inhibitor also works on Aspergillus species?
Yeah, that's another good question. Of course, Aspergillus is a mold, a very different pathogen from Candida. Aspergillus also has a predicted Kix domain, and so we would think that IKIX1 might also be efficacious in that context. We haven't actually looked. So this is something we're planning on doing, but very good question.
More questions?
I was wondering about the second talk about the selectivity of the small RNAs that are being secreted by the plant. Mm-hmm. I mean, do they vary depending on the pathogens you have?
Say it again.
Do they change depending on the pathogens the plant encounters? Because you have to imagine very high selectivity. So how's that?
So I didn't talk too much about this small RNA generation site. I found from both cases the same. For example, from fungal side, they are generated mainly from the LTR regions, retrotransposon regions, and then those small RNAs basically represent a bigger pool of small RNA population, and then during different interactions, different set of small RNAs, some overlap, got selected if they have a positive effect. And then for the plants, it's kind of a similar way. Some are generated from the non-coding RNAs, those TACI mRNA precursors, they are non-coding RNAs. Some are from intergenic regions or transposons, so they provide a large selective pool. But if some has a positive effect, they tend to be stabilized. And then, for example, what we found that those RNA binding proteins help the selection and the loading because, for example, we found the argonauts proteins, especially argonauts-1, and also RNA helicases. So they are associated with rough ERs or MVBs, and then those were selectively bound. A subset of small RNAs got translocated.
All right. I have one more question. Why do you think it is that it's so hard to develop antifungals, right? I mean, for humans, it seems, we have relatively few infections with immunocompromised, but particularly in agricultural sector, I mean, the losses are massive, like economically, right? Why haven't we pushed harder, or is it scientifically not possible?
Because the resistance strain can come up very quickly, because the current fungicides developed to bind to chemicals, bind to specific pocket, then just a couple of amino acid change can totally escape, right? So, but for RNAi actually can circumvent that problem. Even you have many changes along the whole RNA, they can still induce gene silencing. Yeah, I think that's a major reason.
Okay. Mm-hmm. All right, any more questions from the audience? Then I'd like to invite you to join us for lunch and the tech show in the lobby, and we'll reconvene at 2 p.m. All right, thank you so much.